Hello, everyone, and welcome to today's live broadcast, The Importance of Laboratory Diagnostics in the Fight Against Tuberculosis. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by BD. For more information about our sponsor, please visit their site at bd.com. So let's get started. I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time during the presentation. To do so, simply type into the Ask a Question box and click the Send button. If you have any trouble seeing or hearing the presentation, report that problem by clicking on the Ask a Question box located on the far left of your screen. I'd now like to introduce our presenter, Dr. Daniela Maria Cirillo, clinical microbiologist and the head of the Emerging Bacterial Pathogen Research Unit at San Raffaele Scientific Institute in Milan. For a complete biography of our presenter, please visit the Biography tab at the right side of your screen. Daniela, welcome. You may now begin your presentation.
Thank you very much, Susie, and good day, good morning, good afternoon, good evening, wherever you are. And I'm very pleased to discuss with you the relevance of the laboratory for the diagnosis of tuberculosis. I'm very pleased to discuss today. Today is the World TB Day, the day in which all stakeholders come together to celebrate progresses and to really fight for what is still needed to win the battle against one of the worst epidemics that we always had with us, tuberculosis. This is the disclosure slide, and the opinion or whatever I'm going to say is really up to me, and that doesn't reflect the view of the sponsors. And the agenda of today will be, because it's the TB Day, to go through the global burden of TB, to discuss the different programs to stop TB worldwide, the role of diagnosis to eliminate TB, that is actual goal, especially in low burden TB settings, such as the one I am living in Italy, gaps in the diagnostic pathways, and I will conclude with the question for research and the way forward. The global TB epidemic. We have 10 million of TB estimated cases. Out of them, we were able to diagnose a little more than 7 million. That leaves us a gap of 3 million of cases that go undetected in the community, and they are fueling the epidemic. They are spreading the disease. Among the diagnosed and predicted cases, we have approximately 500,000 MDR or rifampicin-resistant cases. Of those, less than a half have been diagnosed, and less than that put on treatment. So we are still generating 1.4 million of TB deaths per year. This is a huge number. When we compare with the data of COVID-19, you need to consider that every single day, 4,000 people die of tuberculosis, and 27,000 get infected with tuberculosis. Those are huge numbers that have been going on and on and on. If we look at the global incidence by sex and country, that really depends on the settings, but globally, the highest burden is among adult men. These figures really changes with the epidemics in the different countries. And it really depends on who in the countries hit by the disease. But in general, women are less affected, although we have 32% that are women and the rest 12% are children. Children are difficult to be diagnosed, so the disease gets in children both unreported or overdiagnosed. And this is really linked to our gap in being able to make a proper diagnosis of tuberculosis in children. If we look at the estimated TB incidence rate in 2019, we still see that the highest incidence is seen in sub-Saharan Africa, where TB epidemic is fueled by the HIV epidemic. Instead, if we look at where the missed cases are in very large number, we can see that of these 3 million of missed cases that are fueling the epidemic, and if we do not find them, we cannot stop the transmission, we have 5 countries that are accounting for the majority of the missed cases. So really implementing the capacity to perform diagnosis in those countries will be able to change the figures of the whole world. The other major problem of the TB epidemic is multi-drug resistant tuberculosis. Multi-drug resistant tuberculosis is defined as the form of tuberculosis that is resistant to rifampicin and isoniazid. We do have good diagnostic tools to speed up the detection of tuberculosis, but of drug-resistant tuberculosis. But of course, in order to do that, you need to have people presenting to the laboratory and to be able to perform the diagnosis. When we look at the number, where do we have the huge problem of tuberculosis is in India, China, and in the country of the former Soviet Union, including Russia. In the European Union, the countries with the highest incidence of MDR remains the Baltic countries. India, China, and Russia by themselves, they have more than 50% of all the rifampicin resistant or MDR TB cases. But the problem may be getting bigger. There are modeling that are based on the projection of what is done today to diagnose and to handle multi-drug resistant tuberculosis that show that in almost all high-risk settings, and here we do have 4 different examples, taking India, Philippines, Russia, and South Africa, all high-burden settings for tuberculosis and also MDR tuberculosis. Continuing the business as usual, we can project that the percent of multi-drug resistant tuberculosis among all incident cases of tuberculosis will increase to more than 12% in India, almost 9% in the Philippines, almost 32% in Russia, and around 7% in South Africa by 2040. Those are huge numbers, and they could really hamper our effort to combat the disease, to fight the disease. Since 1994, the WHO has developed a global strategy to try to combat and control tuberculosis in all settings. They started with the DOTS strategy, and the DOTS strategy was a pretty good strategy that would put the government commitment as the first action. Why is the government commitment so important? If you don't have the money, if you don't have the government that is committed to fight the disease, that allow to have available the resources to fight the disease, really nothing can be done. And then the strategy was case detection through passive case finding. So people self-reporting to the laboratory, and people would have been diagnosed through sputum smear microscopy. So only at that time the laboratory was considered central, but with a very simple technology, more than 100 years old technology. And then all the people that were diagnosed as a sputum positive, they should have been treated with anti-TB drug, with the short course of the anti-TB drug, and making sure that the drugs were always available in the system. So nobody had to stop the therapy because of stock out. In addition, it was the beginning of the establishment of a strong monitoring system, able to perform program supervision and evaluation. It was the beginning of the surveillance program of tuberculosis. One of the best surveillance that was established at the WHO. Of course, that gave some results, but was not enough. So in order, the MDR came out, and it was a huge need to implement the high quality DOTS, not only the direct observed therapy, that is what the DOT means, but the strategy as a whole. And it became clear that two factors were really going to be addressed in order to achieve the results: the TB HIV epidemic and the MDR-TB. And in order to do that, there was no other choice than understanding that the whole health system needed to be strengthened, and everybody should be engaged in the fight against TB. Public and private partnership were at the beginning, and in some countries, they are still the way to go. The private sector is still very important, and the private laboratory sector needs to be upgraded at the public level in order to make sure that they provide the same quality that is provided by the laboratory that are under the NTP system. And then the empowering of people with TB and the community, and enable and promoting research. Without research, we wouldn't be able to have the tools that we do have today to make the diagnosis of TB, and the one that we hope to have in the future. And then in 2016, we moved to the Ntb strategy. The Ntb strategy is really changing the pace, and we are now working around an integrated patient-centered TB care and prevention. So the patient is at the center of our attention, and we need to have policies and supportive systems. No more pyramidal, vertical system, but more integrated system that are able, in terms of the laboratory, even to share platform, to share instrument, and then a big focus on research and innovation. These are the three actions that should make us in the condition to reach the target of 2035. Are we there yet? I'm not sure we are completely on track. We are losing ground. But if we apply those strategies, we should be better suited to reach the goal. Then the WHO is coming with guidelines. I didn't go through all the publications that we are available, but finally we do have what we call consolidated guidelines. We do have consolidated guidelines in all the area, including the laboratory area. And why is so important to have consolidated guidelines? Because guidelines are coming regularly and are updated every year, but in order for people to be able to look at them and really interpret it in the correct way, you need to have one guide that gives you everything, and everything is handy. Otherwise, you may come up with contradictory messages, as in the field of laboratory, in which one guideline was recommending the interpretation of some test in a way, and then other guidelines may have recommended a slightly different interpretation. That could be confusing for the people in the field. But more of all, we do have the very new one on screening. In order to detect more cases, we need to change our capacity to perform screening. We cannot only refer to people that are self-referring for diagnosis because they are symptomatic. We know from surveys that people that are transmitting the disease are non-symptomatic and they are still in the community. With this WHO operational handbook on screening, the WHO is really coming with a roadmap on how to screen, what to use, how to monitor, how to implement, and which group should be the priority target for tuberculosis in order to increase the case detection. Do we pay at all to the disease in terms of the economic impact? Yes, tuberculosis is making everybody poor. Families with tuberculosis, they are suffering devastating costs. And we need to stop the cost of tuberculosis for the patients, coming with more support, including economical support. People don't only die for tuberculosis, they died with their family for the poverty that is increased by the tuberculosis. Another important player in the field is the Stop TB partnership. The Stop TB partnership is the house of more than 1,000 organizations among governmental development agencies, technical agencies, and NGOs. And there is the Mother Stop TB partnership in Geneva, hosted by the UN, but there are also very important national partnerships that are born to help people to fight TB. And we have different examples because the partnerships are really driven by the need of the country. And here you have three different examples. In India, the partnership works with the community to improve TB care, especially for the marginalized and the vulnerable population. They improve the capacity to bring these people to the diagnostic center to make sure that those people are aware of the process and the fact that they needed to be diagnosed if they have any symptoms. In the Philippines, the tuberculosis really brings together the civil society and gives voice to the private sector and to the patients. In Italy, the Stop TB needs to fill the the gap that we have with the engagement of the institution and the government commitment. Government is not perceiving the disease as a major issue, is still perceiving the disease like something that is not an important health problem in our country. So we need to keep the government aware of the problem. In the same way, through the time, the role of patients' association became a huge and important factor, even for the diagnostic field. Patients are supporting and advising, they can share their experience, they can make the changes happening. They can provide the patient perspective, and this is an important factor for the development of new diagnostics. We think that now more than ever, patients should be an important player in deciding what are the priority, the preferences in the new diagnostic field. The Stop TB Partnership has working groups, as you probably know. The working group are the research arm of the partnership. The partnership is a working group on new TB vaccines, on new drugs, and in new diagnostics. And I am the co-chair, together with Morten Rutveld at FIND, of the new diagnostic working group. We do have a vision that is the high quality of diagnosis of TB and drug resistance should be available for all people in all settings. Not only simple tests, but as sophisticated, as sensitive and specific as we could be in implementing the technology into simple devices. As a mission, we foster the development, the scouting and evaluation of the new diagnostic. We try to be the coordinator, the communicator, and the advocacy platform for all stakeholders involved in the field. We do have a vision for what is needed. We need to have triage tests to be used at the point of care, like we are talking now of C-reactive protein for HIV co-infected people. Simple test with low specificity but high sensitivity to rule out the disease and then to really channel into the diagnostic pathway the people that need a better screening. Then we need to have TB confirmatory test that with rapid and integrated DST for the critical drug to drive regimen decision. You know that we are pushing for decentralization of MDR treatment, and for that we need to be tested for drug resistance at the same level. And then we still need to have comprehensive DST, and I will go back to this point when we will discuss the new definition of the XDR-TB. What we have learned from the COVID-19 lesson, if there is something that we did learn, is that we needed to be ambitious. Because with COVID-19, both in terms of vaccine or in terms of diagnostic, we have seen the impossible become reality and in very short time. What was the difference with the TB world? The amount of money that were invested by the whole world in the field. And we need to continue. But the COVID-19 has taught us a lesson. It was probably a good lesson, but we also need to think what is going to be the result of what I call a collision of pandemics. What you see in this graph is the trend in monthly notification of people with TB in 4 high-burden countries, January to June 2020. So you see that the notification, and India has an online system for notification. So the system is really ready to pick up the notification. And what you see is that there was a huge drop in the notification in all the settings. When India was just going up, increasing the missing cases, then a huge drop in the reporting. And that is going to have a huge effect. And what you see on the right graph here is that in India, both the public sector and the private sector were equally affected. And although the numbers are going up, we don't know what will be the results. But modeling analysis shows a possible increase of annual TB death to the number that we had 8 years ago. And the Stop TB Partnership estimates that additional 6 million of TB cases will be notified between 2020 and 2025. And out of them, a substantial number will be children that are the most affected by the disease. So what can we expect? We have seen a really different scenario by this collision of the two pandemics. We have seen the fact that everybody is wearing a mask. Mask is protecting from spreading tuberculosis, but mask is also, and I must say, what is good about COVID and the mask is that mask was stigmatizing the TB patients in the past. Now that everybody is wearing a mask, I've been out with a mask since March last year, so it's more than 1 year now. We feel that nothing is wrong in wearing the mask all the time. Sure, we are hoping to drop that, but still we can live with that. And there are also a lot of diagnostic improvement in the mask. There are studies that have shown that particular strip could be inserted in the mask and could be used as a material to capture both COVID and TB bacilli. So in the future, we could also consider to use the mask as a device, as a diagnostic device to concentrate bacteria, for example, for children that are not able to expectorate. Then social distancing. Social distancing has probably stopped the transmission of tuberculosis in the community. But on the other side, we really have locked the people in their houses, and most people live in crowded houses. And there was a decreased access to care due to logistics, due to the lockdown and other issues. And in addition, our laboratory were reconverted, and the staff was either reconverted as well to COVID work, but was also sick. So we end up being understaffed in the laboratory. And I can share you the experience of laboratory in high burden settings, where the only level 3 laboratories were the TB laboratories. And they were shut down for the TB work and reconverted to the COVID work. So we don't know yet the magnitude of the toll that we will be paying to this epidemic. Certainly, what we know is this. We used to have tuberculosis as the biggest single agent killer in the world. Certainly, 2020 has changed the figure because COVID around the world became the biggest killer. But if we look at the Global Fund eligible countries, that are those that you see in orange here, you see that the COVID-reported TB deaths are less than a quarter of the total death of tuberculosis. So in those countries, tuberculosis is still the major killer. So keep it in mind. Now we need to approach the pathways of tuberculosis disease progression. And this is changing over time. With more understanding of the disease, what we know now more is that we don't talk anymore about latent infection and active disease. We know that we are moving gradually from infection to active disease that is non-symptomatic for a while, or can also go to cycle of disease that go up and down, in which people are completely asymptomatic, not produce, they don't have bacilli, and they move up and down through those phases, becoming infectious only when they move out from the infectious phase. That is an important reflection on the diagnostic that needs to be developed and used for detecting more cases of TB. Here you see a different way to say to show the same different conceptualization of the disease, in which you have the people that are uninfected, people that are just infected, and those are IGRAS positive. They cannot spread the disease. And then you have people that they have active tuberculosis. This is the old and classic conceptualization of the disease. Diagnostic for latent infection, diagnostic for active disease. Now, we do have different phases. We know that people become infected. Some of them will clear the disease, but they will still remain IGRAS positive. Then some of them cannot control the infection, so they will move to the incipient infection. Most likely they are not going to transmit because they are not bacillary, but they have already in their body changes that should be captured in order to make an early diagnosis of this group of people that is fueling the progression, and then is fueling the group of people that will become actively infected and will spread the disease. There is a group in the middle, that is the group of subclinical disease. Those are the people that are totally fine, clinically speaking, but they have already some chest X-ray abnormality, and they do spread the disease. Although they are paucisymptomatic, they could be bacillary producing, and they could really spread the disease. If we care about stopping quickly the epidemic and the transmission, we need to target both the people with active disease and the people with the subclinical, incipient, and latent form of disease. If we look at the cascade of care from the laboratory point of view, we see that we miss the initial diagnosis here, and only if we do a successful diagnosis, we can move these people to treatment without a delay. And then once the treatment is initiated, we need to keep the people in the system in order to make sure that the treatment is appropriate. And the role of TB diagnosis is fundamental and is different, as is shown in this publication, in the different settings. How can we increase the role of diagnosis? In India, the major gap is on the private setting. If the private setting doesn't use the high quality diagnostic, the recommended WHO diagnostic algorithm, then we are going to miss a lot of cases. If we implement algorithms in which we do have the private sector fully engaged, we will be able to decrease the diagnostic delay of 4 to 6 months, decrease in transmission. If we go to Kenya, in Kenya, the problem is the HIV cascade delay that is fueling, is the major gap that needs to be addressed. So, diagnosis of HIV, providing ART, and providing isoniazid preventive therapy will really affect the mortality of the TB patients and will have an impact with a reduction of almost 56% in incidence and 77% in mortality in the future. In Moldova, what can affect the number of can really have an impact in the system is instead the implementation of fast drug susceptibility testing. So implementing almost the the peripheral laboratory proper drug susceptibility testing in Moldova will have the biggest effect in the mortality of the TB patients. And we can see that the diagnostic gap in the TB care cascade are always in the capacity to bring the people into the laboratory and to properly utilize the diagnostic tools as recommended. Consider that we now have a much larger menu of laboratory diagnostics, and not all of them are appropriate for all settings. So right now, what is missing and what should be developed by WHO and partners is a guide to select the proper diagnostic for each setting. There is an evolution of diagnostics for TB. We started from microscopy and culture. We are moving to genomic and universal DST. Consider that when we are talking about universal DST, we are really talking about molecular DST for few drugs. We need to bring this molecular DST for few drugs to a more comprehensive capacity to examine the bacterial genomes and to predict the sensitivity to the drugs that are used for the first-line treatment and for the second-line treatment. And then, in addition, we need to increase the knowledge gap on bacterial host biomarker and to move them closer to the patients with technology that are platform-free. Let me move to one simple example. The WHO has newly defined the XDR-TB. So now the XDR-TB is the form of TB caused by MTB strains that fulfill the definition of MDR, so are rifampicin isoniazid resistant, and are also resistant to any fluoroquinolones and at least one additional group A drug. What is the problem? Group A drugs are bedaquiline and linezolid, in addition to fluoroquinolones. For those, we do have phenotypic and genotypic tests. There are advantages for phenotypic and disadvantages for both phenotypic and genotypic. Definitely, the genotypic is faster. But in order to be applied, you need to know the determinant of drug resistance. Right now, for bedaquiline and linezolid, we are probably not able to report XDR-TB accurately, but not only in high burden and low resources setting, really nowhere, because we don't have very strong and reliable phenotypic test for bedaquiline and linezolid. We are still discussing the critical concentration. We may risk an over-reporting or an under-reporting, and definitely that will take a time that is considered too long to start the treatment of the patient in the periphery. And for genotypic tests, again, we are in the same situation because the determinant of drug resistance to bedaquiline and linezolid are really not as clear as the molecular determinant for other drugs. Let me add more. We do have so few strains that are resistant to linezolid that even the positive predictive value or phenotypic test will be still too low to be interpreted with some accuracy. So we did a survey in Europe to check which laboratory were really able to report, and we find out that even in the European laboratory network, there was really not a large capacity to test for the new Group A drugs. The other major need is the alignment and standardization for phenotypic DST. This is just to show that we have critical breakpoint, defined in the US, defined by the WHO, and defined by Euro, so by the EUCAST, that is the European Committee. We cannot continue to go on with that. We need to standardize and align so the same patient that is diagnosed with MDR with a critical concentration in the US keep being sensitive of resistance if is diagnosed in Europe on a second strain. Whole genome sequencing has a lot of potential. It emerged as a technology that could really revolutionize the way we see TB, as conventional application with fast detection of phenotypic drug resistance, allows us to investigate the genetic diversity, phylogenesis, to determine the lineage. Consider that the more data we are collecting, the more we think that even in antibiotic resistance, we could see a lineage effect. So knowing the lineage will allow us to refine and interpret more correctly the resistance test when it's performed by phenotypic testing. And there's also a lot of public health application in epidemiology and surveillance. ECDC has just established the whole genome sequencing as the way to perform surveillance for cross-border clustering of MDR strains. And then there are unconventional applications that will allow us to study the heteroresistance and the mixed infection, the virulence factor, and to study the new drug-resistant associated mutation and the possible cross-resistance. What is the challenge? The challenge is to move out from whole genome sequencing and move closer to the patient's encounter. In order to do that, what you need to establish as the first action is to have a mutation catalog of all what we know and we consider associated to drug resistance. Only if we know the mutation, we are able to correctly interpret the genomic data that we can provide through whole genome sequencing or next generation targeted sequencing. And in order to have the tool available, we really needed to combine data sets coming from all over the world and to examine to a common pipeline and have a group of experts revising the data and really establish, based on solid algorithm, the association between mutation and drug resistance. And so far we know that some mutations are really confident markers of drug resistance for some drugs, but there are also mutations that we should ignore because they are not associated to drug resistance. And without having this solid background, we may make huge mistake in interpretation. Now let's move very quickly to the subclinical incipient and latent TB, the reservoir of people developing active TB. The current TB infection diagnostic tools are all based on the indirect tests that are only able to demonstrate, either in vivo or in vitro, that there is a host reactivity toward the M tuberculosis antigen. Now the WHO is now coming with recommendation on ruling in TBI and screening different groups. And they recommend both TST or eyeglass to screen high-risk groups and in order to offer them preventive therapy. We do have some new generation of TB infection tests. We do have some new ESAT-6 based skin test that will be coming out. One of them is already available in Russia and in former Soviet Union countries. There are others that are under evaluation and validation, so they will be available and they will have the advantage of the eyeglass not cross-reacting with BCG and other mycobacteria, with the advantage of the TST. So to be very cheap and easy to be performed. And then we have a lot of next generation eyeglasses that are coming in the market. But what is the problem? The problem is that these new eyeglasses will really decrease our capacity to, will decrease the time to result, the cost, but really they will not have a better positive predictive value compared to the current eyeglass. And in fact, what is missing is a good test for the incipient TB and the subclinical TB. We are still very far from that. All what we have right now is a positive predictive value that is too low to be used for that. In reality, the research has been producing a lot of new and very interesting results. So we now have a clear idea of the biomarkers or the fact that the biomarkers are really suitable tests for predicting progression of the infection, for predicting subclinical disease. But unfortunately, when we look at the performance of those biomarkers, they are only able to predict in a very short time the progression to the disease. So they are only able to predict in a time frame of 3 to 6 months. And this is what we have today. So if we are only able to predict disease progression between 3 to 6 months, we should be testing the people too often and become unpractical. So there was a recent and very interesting publication in Lancet Respiratory Medicine that really focused on that. they were looking at 17 different signatures that may come in the market in the near future, and they were coming up saying that no one of them is really meeting the target of the technical product profile established for those tests by the WHO. What is interesting then to see is that most of the signatures converge in the target. So continuing the research is very likely that we will be able to get a suitable test in the future. What are the priority research questions for the future? What is the impact of the subclinical incipient TB in the transmission dynamics? We need to have a test to detect this status in order to find out. What will be the performance of the host biomarker for the different status of disease? Do we need new tests to stop therapy and declare cure? We continue the therapy for the same amount of time for everybody, but we know that someone could stop earlier and someone could stop later, and the culture is not the right proxy. Universal DST. How to cope with capacity and logistic all over, and then tools based on either genotypic DST and phenotypic DST for any new drug entering in the market. We are tired to see the drug before we have the suitable test. And now this has been one very difficult year. But in this very difficult year, the society that I'm honored to represent, the European Society of Mycobacteriology, has been working a lot in the partnership with Becton Dickinson to provide training and to provide webinars that could be of interest for the majority of the people involved in the fight against TB. So we are going to host 3 different webinars at the end of May, beginning of June, on sample best practice for a successful TB diagnostic, non-tubercular mycobacteria for diagnosis to treatment, how to integrate the diagnostic offers into a suitable algorithm in different settings. Speakers will be excellent, and I really invite all of you to follow the European Society of Mycobacteriology website to be aware of the date and the time. And with that, I'm reaching my conclusion. Despite progresses and gradual decrease of global incidence worldwide, TB hits hard, still hitting in many countries in the world. The larger menu of diagnostic available today needs to be supported by a guidance on how to perform choices that are proper for each setting. Universal DST concept will be met by application with user-friendly genomic tests. Current tests to Ntb will be missed. We need to develop more to work more. We need a test for incipient TB with a sensitivity that is acceptable for at least up to 6 months. Persistent tests will be needed to apply the test and treat strategy to eliminate the infection. The current IGRA are not going to solve the problem in high burden settings. What the true impact of COVID-19? I don't know. That will only become clear in the next future. And I need to thank you for your patience. I'm open to all your questions. I also would like to thank my collaborator, the people affected with TB and their family, all the people that are involved in the fight against TB from all aspects, from scientists to companies to society members, and every one of you. Thank you very much. And now I am happy to tell you that, to remind you that today is the 2021 TB Day. Let's hope that next year we'll meet in person. Thank you very much for your patience.
And thank you, Daniela, for that informative presentation. And we will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's get started. It looks like, Daniela, we already have some great questions coming in from our audience members. Our first question: how can we find out more about the educational series by ESM?
You should visit our website, the European Society of Mycobacteriology website, and you will find in more or less 2 weeks the exact date and time. We also have on YouTube the older series of webinars that you are more than welcome to watch, and you'll find the link on the website. As well as you are more than welcome to look for the date of the meeting that will be at the end of June, beginning of July. We will have an online meeting with almost no fee for registration.
Thank you so much. Our next question is from an audience member who is located in Silmar, California. And she says, I'm located here in Silmar, California, a historically significant location in early 20th century of TB treatment and research. And I've also worked with Dr. Froman in 1979. Is there a clear link to TB infection by smoking, as there is a connection with HIV?
Yes, there is a link between smoking and TB infection and progression of disease and also outcome. And the link is coming clear from a lot of publication that shows how the deterioration of the lung promoted by the smoking, by the heavy smoker, is really is promoting the progression of TB from infection to disease, with form of disease that could be more difficult to treat.
Thank you so much. And again, I want to thank our audience members for these great questions coming in. Our next question: how would you recover the bacillus trap in a children's face mask to work as a collection instrument in diagnostic?
This is still a prototype that I have seen, and there is a special material that is it's buffered, that while you are exhaling, because we are now wearing the mask all day long when we are outside and at work, so you keep exhaling and you can exhale the bacteria get trapped, and then you can dissolve the bacteria and get the, certainly you cannot use a smear microscopy, but you can have different technologies to detect the bacteria and that are trapped on these strips.
Thank you so much, Daniela. And we have time for a couple more questions. Is Tbt Cell Response still a valid tool to support TB programs from who?
From WHO.
Yes.
Sure. You know, ELISpot was never completely, ELISpot is endorsed, is still available on the Global Fund menu, but so it can be purchased to the Global Drug, the GDF. Definitely those tests, as well as the skin test, the TST, and the new skin test will go through a revision by the WHO. Those are very imperfect tests with the problem to have no capacity to predict who is going to progress into the disease. So for the WHO really focus on the high risk category, and whoever has a positive test will need to be treated. And in fact, when we talk about children, we think that the recommendation are that small children should be treated if they are contact of a case. Anyway.
Daniela,
but definitely other tests with a higher predictive value for progression are needed.
Daniela, we have time for one more question, and this is a good one. Is there any vaccine against TB?
Well, you know that there is the old BCG is still in use and is an important vaccine for children. Really is able to prevent TB meningitis in children as well as a serious form of TB in small children. Doesn't protect adults from pulmonary tuberculosis later on. There are several trials with several promising candidates that are ongoing. But I must say that one of the effects of COVID-19 is that all the trials have been either stopped or delayed. So the COVID-19 will have a huge impact on research, and will only also have a huge economic impact, because what was the cost of a trial has been increased because of the stop caused by COVID of about 10 to 20%, with no money, leaving the scientists without money to complete the trials in some cases. But yes, there are promising candidates.
Daniela, I want to thank you again for your time today and for your important research. And I'd also like to thank LabRoots and our sponsor BD for underwriting today's educational webcast. And before we go, I want to also thank our audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And
Thank you very much, Susie. I will be happy to answer the question if people would like to email me. That's not a problem at all.
Awesome. Thank you so much again, Daniela. And you can view this webinar on demand, and LabRoots will alert you via email when it's available for replay. That's all for now. Take care, everyone, and stay safe. Until next time, bye-bye.
Bye-bye.